This is the Chemistry, Biology, and Math Revision Hub. In this video, I will be discussing the January 2022 Biology Unit 6 paper. This paper has a total of 50 marks as well, so I will begin with question 1. Question 1 says, the photograph shows part of a field in Kenna plant hibiscus cannabinus. So it says crops of Kenna plants are grown throughout Asia. Several products are extracted from these plants. Oil can be extracted from the seeds, and this oil can be used as a biofuel. This is important if something can be used as a biofuel. And then they're going to say fibers can be extracted from the stems and used to make suitable or sustainable material. Sustainable material, again, from your AS biology, you remember you studied about different types of fibers and their uses. So here I marked the key important parts here, the oil that can be used, oil from seeds that can be used as a biofuel, and the fibers that can be used to make sustainable material. Let's go to the first part. It says, describe an experiment to measure the tensile strength of fibers that can be extracted from these plants. So if you want to measure the tensile strength of fibers, you have to make sure the fibers extracted from the different plants are going to be of the same size. If it's length, let them be of the same length, let them be of the same thickness, and if possible, let them be of the same mass. The only difference is that these fibers are from different materials. So you keep all the other variables constant and only concentrate on the different types of materials the fibers have come from. So in describing an experiment, we always begin by stating the dependent variable. Remember, the dependent variable is something that you're going to measure, so you need to tell the examiner what you're going to measure. In this case, I say the dependent variable is the mass required to break the fiber. Basically, we are measuring the strength of the fibers by looking at how much mass we're going to exert onto them in order for the fiber to break. So the result from this experiment, or the measurable result, is going to be how much mass this fiber can take before it can break. So here you say the dependent variable is the mass required to break the fiber. Here you get 1 point. And then, like I said already, use fibers of the same length and diameter. If they are the same length and diameter, it's going to be possible that they're going to be of that. Basically, they are equal in size and everything. Then we will clamp the fibers. I'm going to show you something from your practical book before I come back to that part. This is how you're going to clamp. There is a retort stand here, and then so 2 retort stands are going to be here, and then there is a clamp. On the first retort stand, we have a clamp, and on the second retort stand, we have a clamp. So what happens, we position the fiber in between the 2 clamps, and then we'll position weights onto this fiber and observe how much weight this fiber can take up before it can break. This is going to be the setup of the experiment. However, in this question, you are not required to draw this. I just put it here so that I can be able to demonstrate to you. So I will go back to the clamping. So we clamp the fibers between the 2 clamp stands, and then we will add known masses to the clamp fiber until it breaks. You have to add until it breaks. So the fibers should be of the same age. Of course, we already know if they're from the different materials, same size, same length, they have to be the same age. And since we know that some fibers, due to humidity, their properties can change, you have to make sure the place where the experiment is being carried out has the same conditions, same temperature, same humidity. And then lastly, we can calculate the tensile strength by dividing the force, which is the mass added basically, by the cross-section area. Here you will repeat the experiment under similar conditions and calculate the mean. Always, this last part of repeating the experiment to calculate the mean is going to be very important if you want to score marks. Now, you did not have to write all these points in order to get 6 marks. As you can see, 1, 2, 3, 4, 5, 6, 7. There are 7 measurable things. I know some students would have failed to put these, but that doesn't mean you wouldn't have got the other marks. So this is how you would answer this question. I'll go to the next part. It says, cellulose is a major component of plant fibers. Describe the structure of cellulose molecules. Now, cellulose molecules, cellulose is basically made up of beta-glucose molecules, and these glucose molecules are joined by, so basically one glucose molecule is joined to another glucose molecule and then another glucose molecule, making a polymer. So again, cellulose is a polymer made up of only beta-glucose molecules. These molecules are joined by only 1,4 glycosidic bonds. So this is a keyword as well. And then every glucose molecule in the chain is inverted. So if one is like this, let me say like this, the other is going to be inverted in the other direction. This part is not going to be facing down. It's going to be facing up like that. Again, if you go back to the structure of cellulose, you will see that one group is going to be, what if the first molecule, the first glucose molecule is going to be, if it's facing upwards, the other glucose molecule is going to be kind of inverted like that. And then the next one will be the right way, the other inverted continuously. And the chain produced is not going to be branched. So it's an unbranched chain of a molecule that is going to be produced. The last part says, give one reason why the use of biofuels can reduce the effect of climate change. Now, biofuels usually are going to be generated from plants, of course. So if they're generated from plants, these plants would have taken up carbon dioxide in order for them to grow, of course, photosynthesize, produce their own food, and be able to grow and increase in size. So it means all the biomass present in this tree is mainly due to the carbon dioxide this tree took up when it was growing. So it means when we use biofuels, we are burning and releasing carbon dioxide that was already taken away from the environment, which is better than when we use fossil fuels that just add up something that was not in the environment. So here I said, because on combustion, the biofuel only releases carbon dioxide that was absorbed during photosynthesis or during its growth. I will continue to question 2. Question 2. The photograph shows our garden snail, Helix asperser. This species is endemic to Western Europe. So we can see the snail here. And the next part says, snails feed on plants in warm, moist conditions. In dry conditions, snails seal the entrance to their shell with a membrane. The photograph shows this membrane. This is the membrane you can see here on that surface. It has sealed the entrance to where the snail is. I will go to the next page. So this says, student investigated the volume of water needed to stimulate snails to break through the membrane at the entrance to their shell. The student used the following method. A group of 15 snails with membranes was used in the investigation. So 15 snails were used. The diagram shows how each snail was held upside down. So the snail here, as well as the membrane and the support. Each snail had 2 drops of water added to the membrane every time. The time taken for each snail to break through the membrane was recorded. Part A says, suggest one reason why the student thought it was ethical to use snails in this investigation. This kind of question is common, not only for snails, but other, like worms and other organisms. So when they use organisms, it means they do not feel much pain. It means they have a simple nervous system, and therefore the pain is not going to be felt like other complex organisms that have complex nervous system that can feel pain greatly, or a lot of pain when they're in specific positions or when they use for experiment. So the student thought that the snail does not feel pain since they have simple nervous system. This is usually a model answer for any kind of question asking about ethical issues regarding organisms used in experiments, mostly simple organisms. Okay, let's go to the next part. It says the table shows the results of this investigation. So here we see the number of drops of water added. Here they added 40, 80, 120, 160, and 200. And then the total number of snails that broke through their membranes, the results are shown as well. So they said the mean diameter of each sphere drop of water was 3.0 millimeters. Calculate the volume of water added to a snail in 1 minute. The volume of a sphere can be calculated using V is equal to 4 over 3 pi r cubed. So remember, again, some students actually fail to remember that radius is equal to diameter over 2. So if you're given the radius, you have to divide if you're given the diameter, you have to divide it by 2. Since this diameter is 3.0, radius should be 1.5. After knowing that, the rest is going to be easy. So volume is going to be 4 over 3 times pi times 1.5 cubed. And the answer comes out as 14.137 times millimeters cubed. However, they've told us the final answer should be in 2 significant figures. So, and again, one key thing, this is not the volume, this is the volume per drop. We need to find the volume in 2 drops. So I have to multiply this answer by 2, as you can see here, and obtain 28.28274 into significant figures, it's going to be 28 millimeters. If you want to get some marks, maybe usually questions requiring you in biology, questions requiring you to write in significant figures could be many. So you have to try and make sure you understand the little mathematics regarding biology. The next part says, give two abiotic. When they say abiotic, it's kind of non-living variables. So give two abiotic variables other than the independent variable. The independent variable is something that is varied or something that is changed during the experiment. So that could affect this experiment. This experiment is involving snails, so light intensity, the pH of the water, the humidity of the room, the temperature of the room, all those could affect the behavior of the snails during this experiment, and they could lead to having erroneous results. So part 2 says, name one of the variables you have identified in one. I used variable, which is pH, and then they say state how this variable could be controlled. We control pH using a buffer. If you had chosen temperature, then you can say use a thermostatic control, water bath, or sorry, thermostatic control. In this case, because snails are not in water, so do not say water bath. You say thermostatic control, maybe in a room where there is the temperature is going to be thermostatically controlled. This part usually works. This part here works usually if substances are going to be dissolved in water or in an area where water can be able to come in. So you can use a water bath. Next part says, I said humidity. For humidity, we can use a humidifier or a dehumidifier. And light intensity, of course, you have to use a bulb at a specific distance in order to control light. The next part says, suggest the effect that not controlling this variable would have on the results. Of course, the results will not be valid because in this case, change in pH would endanger the enzymes, or would affect, let me just say affect the enzymes that are potentially participating in the breaking of the membrane, and therefore the membrane may not break efficiently, or it may not even form efficiently. So that could affect the experiment. This part says the student decided to repeat the investigation using the same apparatus but adding 4 drops of water every time. There was no significant difference in the results when the number of drops added each minute doubled. Suggest why this did not have an effect on the time it took the snail to break through the membrane. If you look at this, it tells you like it was not about the volume of water, or how much water has been added within a short time. So the only thing that led to the opening is that water is present. So I say because it is presence of water and not the volume of water that was important. The volume of water, not the rate at which the water is actually being added. So presence of water was important, but how much water is there or the rate at which the water is being added was not important in determining that the membranes break. Again, remember, this is a physiological process. If a physiological process is going to occur, it means you might see it outside. However, it's being controlled by many mechanisms are working, or many processes are being carried out within the snail that lead to the formation of this membrane. So maybe you would add water. However, the enzymes have not been activated because the temperature is not appropriate, and therefore the membrane cannot be formed. So it's more than one factor that leads to the formation of the membrane or breaking of the membrane. So here I say it probably another factor limits the rate at which the membrane breaks down. So something else, meaning it could be multifactorial. This brings us to the end of question 2. Question 3, the photograph shows a species of mayfly nymph. Mayfly nymphs live in streams and rivers on every continent except Antarctica. Many species of mayfly nymph are found in unpolluted streams. All mayfly species have a 3-pronged tail. There are fewer species and lower numbers of each species in polluted streams. A student read a report that low levels of chlorpyrifos, an insecticide, may have entered some streams. The student used the method shown in the diagram to collect mayfly nymphs. Samples were collected from unpolluted stream A, of course, so here unpolluted stream A and unpolluted stream B. So we're going to make a comparison and see if there is a significance basically. So this is the mayfly nymph they're talking about, and let's go to the first part of question 3. It says, the diagram shows kick sampling in a stream. So you can see how the stream was sampled. Basically, the person is dressed well. They're putting on boots on their feet, and they're trying to. The stream is flowing down, but they're collecting of course, the biological species they're going to use for this experiment. So the number of mayfly names from each kick sample was recorded, and the students' results are as below. So here we see there is for stream A and for stream B. These are the raw data, or this is the raw data that they presented. The first question says, state the null hypothesis for this investigation. This is a comparison. So if we know that in some part of the water there is pollution and the other there is no pollution, it means we are trying to show that there is no significant difference between the mean number of mayfly names in stream A as well as stream B. So this is determining if there is significance between the mean numbers, not just the numbers, the mean numbers, because, you know, experiments have to be repeated and the mean have to be found so that we can determine significance using the mean number. Please learn how to state the null hypothesis, because this question comes almost in every Unit 6 paper. They'll ask you to state the null hypothesis. So here they say, draw a suitable table to display the raw data. If they say raw data, it means show it the way it has been presented. And your calculated means for stream A and stream B. So this is my table. As you can see, let me try to make it kind of smaller so that you can see all of it in one slide. Okay, like that. So the number of mayfly names, we have stream A and stream B. I showed everything as raw data. So you will, remember they told us there were 15 samples. So you will add up everything until here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So there are 15. Get the total, divide the total by 15, and find the mean. For those of you who do not know how to find the mean, please revisit the basics of math are required in biology. So we will do the same thing here, add up everything to here, and then you divide by 15, and then this is what you get. So those are the main values we were looking for. And remember they told you show the raw data, so data has to be shown exactly as it was presented in that right order. I will continue to the next part. The next part says, again, I'm going to make it smaller so that you guys can see my graph. They say plot a graph to show the mean number of mayfly names for stream A and stream B. Include an indication for variability of the data. When they say variability of the data, it means you have to show the error bars. Error bars are this. You guys can see the upper bar, the lower bar. If this is the mean value here, this is the mean value, the highest value is that and the lowest value is that. So we put a line to connect the highest and the lowest. That is what we call the error bars. For this one, we have also done the same. You can see the lower and the upper, and then connected through the mean to the lower part. Those two are error bars. Now, we will compare the lowest number, the highest number, as well as the mean. So here on the x-axis, we just put stream A, stream B, and you have to put, of course, the labels, which is variety of streams. On the y-axis, we see the mean number of mayfly name. The scale has to come from 0, so you have to find your appropriate scale and draw. Remember to put units. And then this is easy. This is just a bar graph you're required to draw here. So the next part says, the student analyzed the data using the t-test with the formula. This is the formula given. So where they were really kind, they've told you all the information. So where x bar means the value for each stream, and n is the number of samples for each stream. SA squared is 116 and SB squared is 116. This is kind of easy because you're just given all the values. This is given, which is that is given. The whole thing is given. That is given, the other is given. This one we know it's 15 and that is 15. So this value here, it's of course we know it's going to be the mean value. So we have one is 36 like here, and the other is going to be 32. From our table we drew, so we just fit everything in here and then use our calculators to find the answer at the end. So I got the answer as 0.933. This is your calculated value using the t-test. And of course, they're going to, so for me, I used 0.933 because it's the answer I got after my calculation. Let me continue to the next. So here they have given us the table of results, basically. So they say the table of the table shows the critical values of T for different degrees of freedom. Now, the degree of freedom is found by getting NA minus 1. So it's going to be 15 minus 1, giving us 14, and the other side 15 minus 1, giving us 14. It means the degree of freedom is going to be 28. So you will come to your table of critical values and look at, you will go through, in biology we do at P is 0.05. So in P is equals 0.05, you will look to where 28 is. This is the degree of freedom. So the value where these two meet, that is the value you're looking for, and that is going to be the critical value for this result. So our critical value is 2.05, yet our calculated value is 0.933. You can see at 28 degree of freedom, the value 0.933 would lie way below 2.05. So it's going to be below or lower than the p value at 28 degree of freedom, which is critical basically. This is the critical and this is the calculated. The calculated value is lower than the critical value. So I will continue to the next part. They say, comment on the results of this investigation. Use your graph and the information in the table to support your answer. I calculated this, as I've already done before, and I got 28. So I say that 28 degree of freedom, the critical value at P is 0.05, is 2.05. And then I say, since the calculated value, 0.933, is less than the critical value, we will accept the null hypothesis. This is very important, knowing when to reject and when to accept the null hypothesis. And then lastly, remember they're asking you to comment. I said, so there is no significant difference between the number of my finding in stream A and stream B. So they are not related. That is how you conclude on this result. I will go to the last part of question 3. They say, explain why it may not be reasonable to draw valid conclusions from the results of this investigation. If you guys would see any Unit 6 biology paper, it's like they have the same pattern. It's easy to, if you have done various papers, it's easy to predict the questions that are coming, and it's easy to be able to get a higher score. So here we know. Remember, we looked at the error bars. The error bars did overlap. I'm going to take you back slowly so that you can see what I'm talking about. And again, please excuse me for this. I'm going to take you back slowly to my graph, the graph I drew. Yes, this one here. If you compare the two results, look at these error bars here for this one and that. You can see they are overlapping. So because the error bar for stream A and for stream B are overlapping, it means our results may not be that reliable or they may not be that valid. So for my first answer, I say the data is not valid since the error bars overlap. Remember, again, here they're asking you why it will not be reasonable to make a conclusion that actually there is no significance, as we have concluded previously in D. So it's possible that other factors like flow rate, temperature, pH, light intensity, or the pollution degree of pollution was not monitored properly, and these can affect the results. So in some areas, it could be because they did not put into consideration or they did not put into account all the other factors. And last, they say the sample should have been taken one more time on another day or on another day, basically, or at the same day, at least another time during the same day to make comparison. One time per day may not be that suitable to make valid conclusions. This is the end of question 3. I will continue to question 4. Question 4, the photograph shows yeast cells. In suitable conditions, yeast cells respire and multiply. A student formed the following hypothesis. The higher the temperature, the faster the rate of respiration. Plan an investigation to test this hypothesis. Your answer should give details under the following heading. So in this case, actually, I can just remove this. We are required to plan an investigation. If you wanna carry out an experiment to determine the rate of respiration, usually in respiration, you guys know, respiration oxygen is used and carbon dioxide is produced. However, when you use a respirometer, sometimes it's really going to be harder for you to distinguish between the two gases. Now, since we know that carbon dioxide is going to be released, we can use soda lime to take out the carbon dioxide and then we will measure the volume of oxygen used up, and that can be a suitable way to ensure that we have only one gas present at a specific time, and therefore any changes within the respirometer are due to the oxygen that is being used up. So in my explanation, I actually used the volume of oxygen produced to determine the rate of respiration. So I will go straight to the first part of question 4. It says, describe preliminary. When they say preliminary practical work, this means before you do the major experiment, you have to know something. An example, you have to determine which species are you going to use, how old should they be, what conditions are you going to use, what temperature are you going to use, what is going to be the pH, how many species are you, how many different organisms are you going to use, what volume of oxygen do you need. All those things are determined in the preliminary practical work. So here when they say describe preliminary practical work that you might undertake to ensure your proposed method would provide quantitative results, you have to find a suitable temperature range for the experiment in order to obtain representative results. We have to find a suitable way to measure the volume of carbon dioxide produced or volume of oxygen used. And again, I used volume of oxygen used because it's easier. Determining volume of carbon dioxide produced could be kind of harder in comparison to that of oxygen. And then you have to find the suitable number of cells. These are the cells, yeast cells or mass of yeast that is going to be used for this respiration. Again, this is not the only thing you're going to determine the preliminary practical work. You might end up doing more than this, but at least you had to put very significant things. In this case, I put temperature because temperature can govern the rate of respiration. And then I put, of course, measuring the volume of oxygen. That is how you know that dependent variable. And then, of course, the mass of the yeast, because this one, the number of yeast cells that are present is going to greatly determine how much volume of oxygen is used up or how much carbon dioxide is going to be produced. So the next part says, devise a detailed method including how you would control and monitor important variables. Here again, you begin by stating which one is the dependent variable. I say the dependent variable is the volume of oxygen used up per time. This one here gives you 1 mark. The volume of oxygen used up because, again, like I told you already, I used oxygen because it's going to be easier. You will just monitor the position of the colored solution in the respirometer, and then you can know how it has changed. I want to first take you to the image I have down here, and then we'll come back to explain. This is a simple respirometer. You can see we have soda lime down here, and the organisms are on top of the gauze. Let me take that away. These organisms are respiring, and as soon as the carbon dioxide is released, the soda lime is going to take up that carbon dioxide so that it does not come into this area. So in this area, we are going to have only oxygen, and therefore within this whole tube, it's going to be only oxygen circulating in there. We are trying to not mix up the two gases within the same area, so the soda lime is going to take up all the carbon dioxide. So in here, we're going to have oxygen as well. So we will release the oxygen into here. Make sure this dye is in this position or the color solution is in that position, and then we will close this tap. When we close the tap, the organisms will begin to respire, and when they respire, they are using up oxygen, so the color solution is going to move this way. Of course, we allow it to move, and remember we have the scale. When it moves, maybe the experiment can be ended when it has moved a specific distance. You can measure that distance and use this formula. V is pi r squared h, volume of a cylinder. That length is going to be h, and then you know the diameter of that tube. So you can use it to find the radius and therefore determine the volume of oxygen used up. Or you can determine the volume of oxygen used up by refilling after this dye has come to this position, you will use the syringe to refill and take back the color solution back to its original position, and then you will be able to know how much volume of oxygen has been deducted from the syringe, and therefore predict that will be actually the real volume of oxygen that was used up by the respiring organism. So it's really easy to do this. Now that you have understood how the real experiment looks like, I want to take you to the description. So the next part I said, add these cells to different solutions, of the same concentration. Remember, they're going to be in different solutions of the same concentration maintained at 5 different temperatures. In this case, we are determining the rate of photosynthesis, the rate of respiration and how temperature is going to affect it. So the added yeast should be of the same species, age, and mass. Yes, this is a variable we're trying to control. And then we will use a respirometer with soda lime to remove the carbon dioxide produced. Then we will determine the volume of oxygen by refilling the respirometer with oxygen from the syringe to reset the position of the color solution. You could do this, or you can do the V is pi r squared formula I've told you, whereby you just measure the distance, and then you would get the pi as well as the radius and find the real volume of oxygen that has been used up. Next, you control pH. Of course, the pH of the buffer solution has to be controlled. The pH has to be controlled using a buffer solution, and then the concentration of glucose used has to be controlled. If the small organisms are using glucose, the glucose concentration has to be the same in every repeat experiment you carry out, and then we repeat the experiment at the same conditions and calculate the rate of respiration by dividing the volume of oxygen produced by the time it took the experiment to be carried out. That is how you determine the rate of respiration when the temperature is being varied, basically. So let's go to the next part. They say, describe how your results should be recorded, presented, and analyzed in order to draw conclusions from your investigation. So for me, I say, you have, after getting the results, of course, it's going to be temperature and then the rate of respiration. We expect as the temperature increases, of course, the rate of respiration is going to increase until a specific temperature that is going to be higher to cause denaturation of some enzymes, and then the rate is slowly going to decrease. That is predictable. So what I did, you have to say, you do not necessarily have to do that, but you can do this here. So they say, I say, draw a table of raw data with headings and units. You have to do that. The results you have got from the experiment have to be tabulated. The tables should have headings as well as units. And then we should calculate means. Remember, you're carrying out, let's say at 5 degrees, you do maybe 3 experiments. At 10 degrees, 3 experiments, so that every result you write in the final table is going to be a mean value in order to ensure that your results are valid. Then we will draw a line graph with axes and labels. It's like a scatter diagram, and then we will use an appropriate statistical test. It could be Spearman's, it could be a t-test, it could be anything, so that correlation of the results can be established. And once we do that, we can draw conclusions to determine whether the rate temperature affects or it does not affect. The last part here we have is they say give 2 limitations of your proposed method. Of course, there are going to be other factors. In this case, our proposed method is not going to be, so it's going to be difficult basically to prevent contamination during the whole culture. So there is a possibility that other microorganisms could be respiring as well, using up oxygen if they are aerobic, meaning the volume of oxygen used up is going to be kind of more, and it's not due to our organisms, but it's because of the contaminants that are going to be present in there. Another thing is it's going to be difficult to measure small values of change in the dependent variable. Remember, if you're measuring the volume of oxygen used up, and let's say it's really, really little, it means it's going to be very, very difficult for you to do that. This is about 2 marks, but I added in something here like carbon dioxide could dissolve, of course, in the liquid. Remember, we put these organisms in a solution, a glucose solution. So carbon dioxide can dissolve in the glucose solution, and that means, or basically, or any of the gases could dissolve in the glucose solution, and if we cannot measure them, then that is going to be difficult. Yeast can possibly be both aerobic and anaerobic, meaning after producing carbon dioxide, it can also use that carbon dioxide in anaerobic respiration. So those are all setbacks. This brings us to the end of this paper. Don't forget to subscribe to our chemistry, biology, and math revision channel. I will see you in the next video.